Hello and welcome. We're going to take a look at Unit 2, Lecture 5, called Intro to Genetics. And we're going to have two lectures this week about genetics, and we're starting off with a broad overview of the topic. So here what I want to try to do is give you an understanding of kind of what genetics is and what's studied. We'll go a little bit more into some of the structure of DNA, which is important to kind of understand how it works. And then we'll end with a brief note about how DNA replicates, which is another key part of really how life works in general. So starting off here with an overview, it says genetics is a branch of biology concerned with the study of genes, genetic variation, and heredity in organisms. Heredity is essentially how you pass on your DNA from one generation to the next. So we start with just the study of DNA, which usually involves the study of individual units called genes. In the human genome, for example, we have roughly 19 to 20,000 different genes. So you could study all of the DNA, which is hard to do, or much easier is to study one gene at a time. And that's very often what geneticists do, is they look at DNA in kind of individual units called genes. Now, another key thing is to then compare genes from one organism to another. That could be to humans, or that could be comparing humans to other organisms that are either similar or not alike at all. And so here, very often, you're looking at differences in DNA sequences. And so those differences help account for differences in what we see at the macro level, the broad level. And then from a hereditary standpoint, geneticists typically are looking at how you pass on your DNA. Is it always the same? How does it differ? How does offspring resemble yet differ from parents and things of that nature? So that's kind of the idea of heredity. Now, these can get a little bit more specific. So going a little further in depth, one of the key things that you have to really understand about DNA is its structure, which then relates to its function. So, for example, DNA is made up of building blocks, which we'll get into here in a minute. And these building blocks, called nucleotides, are represented by the letters A, T, C, and G. So how those letters group together gives us the function, or rather the structure of DNA, which then leads to its function. Understanding function, you have to understand structure. In the next lecture, what we'll look at more in specifics is the expression of genetic traits. And so we'll refer to these terms transcription and translation. This is essentially how DNA operates, almost like a system, almost like a computer system, where you store information in the computer, but then you access information by clicking buttons and accessing programs and things of that nature. DNA works in a very similar kind of way, interestingly. So when we express it, we are looking beyond just the information. What is the information doing in the DNA, and how is that used in a cell? So that's what we'll look at in the next lecture. Another aspect is the change of genetic material over time. I won't lecture over this as much, but you will see this in the textbook review. So this is often centered around the conversation of mutations. How does the DNA change based upon mutation in the DNA? Another conversation about that is called epigenetics, and I will mention that at the very end of the next lecture. Epigenetics is a newer branch of genetics, and it kind of looks more at how the structure of DNA changes rather than the information in the DNA. But one way or the other, DNA changes. It never really stays the same, and so that helps us understand why organisms change and adapt over time. So going a little bit further, when we study DNA, the study can be done at different levels. So, for example, we can just look at the organism as a whole, and we can try to look at how the DNA relates to the entire organism. This is often referred to as organismal genetics. And this is really how the entire field started, well over 100 years ago. Scientists and investigators, naturalists as they used to be called, were able to kind of figure out that organisms had inherent traits that were passed on. And so you could kind of look at the whole organism to figure out kind of what was going on there. Now this has changed over time. to where scientists then were able to relate genetics down to the cell level. They realized that in a multicellular organism, for example, like humans, that each cell was actually different, despite the fact that they all had the same DNA. So now you're looking at kind of why each cell is different, even though it has the same DNA, for example. Now, as scientists progressed further, they were able to then deduce that actually the DNA is made up of units called chromosomes. And chromosomes are kind of like large units of DNA. In humans, for example, we have 46 chromosomes. And sometimes you say 23, so what we actually have are 23 unique chromosomes and then 2 copies of each of those 23 unique chromosomes. So in other words, we have two copies of 23 chromosomes. So sometimes you'll see it as 23, sometimes you'll see it as 46. But those are kind of the large segments of DNA divided up within the total. Now, today, where most DNA analysis takes place is at the molecular level. And what this basically does is it gets down all the way to the most specific aspects of DNA, the molecule of DNA within a cell. And this gives us the most precise level of scientific investigation. So while technically all of these levels take place today, most of the emphasis is at the molecular level, which is where you get the most specific information at the DNA molecule. So that's kind of the way that you can look at genes and genetics from a broad all the way down to a specific level. So when we talk about genes, very often you hear this term, people kind of understand to a certain extent, genes are what make you who you are. You have good genes and bad genes and things of that nature. And we can kind of understand these from 2 different perspectives. Early on, as I mentioned, scientists were studying life, and without really knowing exactly how it worked at that time, they were able to deduce that organisms had fundamental characteristics that were thought to relate to some kind of information in the cell. And so from a classical standpoint, we look at that today and realize that that's still largely accurate. Not entirely, as we'll see here in a second, but the idea here is that the fundamental unit of heredity responsible for a given trait can be traced back to a gene. Okay, so in other words, how something looks is related to its genetic information, and that's kind of the classical view of that. So, for example, take a simple visual concept. If you have freckles, you have a gene that codes for freckles. Never quite that simple, by the way, but that's sort of a way to think about it. So in other words, you could see a freckle or freckles, and you could deduce that that comes from a gene in a theoretical kind of way. Or if you don't have freckles, then you lack that gene or you have a different gene. So something that you see is related to something in the DNA. Now, while that still holds true, it's not the full picture. So to get more specific, scientists have realized that there's a more sort of modern take on that, what I call here kind of a molecular biochemical view of a gene. And what this tells us is that genes actually code for information for a cell function. And it is not the case that all cell functions give visible, observable characteristics. So in this view, what we see is that we have DNA splayed out over a long segment, and then a little piece of that segment called the gene ultimately codes for a cellular function. And very often, these cell functions are not visible. They might code for a cell receptor or a hidden cellular trait that you would never actually observe directly. And so now scientists can use that to kind of realize that it's not always something observable like a classical view. These often relate to hidden cell functions that don't really manifest unless you study them really, really in depth. So that's kind of a more technically accurate take on it, although both still hold true to a large regard. Now, when you want to get even more specific, what a gene technically does, generally speaking, is that it codes for a certain protein or RNA molecule. And I've talked about the central dogma of biology several times, where I show that DNA converts to RNA, which converts to protein. And so what we're showing in that central dogma figure is that ultimately DNA holds the coding for how to make RNA, which then very often is used to make protein. Not always, though. So in other words, genes code for RNA and or protein, and that's more specific. Now, what the RNA and the protein do then relate to things like observable traits or cell functions. So you're kind of getting all the way back down to the core essence of it, that while these do create characteristics, they are ultimately based in protein and RNA. And that's kind of what this is saying here. Now, when scientists try to describe these phenomena, they use two different terms, genotype and phenotype. When you use the word genotype, you're generally referring to the genetic information or the genetic makeup of a cell. So sometimes you'll use the word gene, which describes individual characteristics or individual coding segments, or in some cases you'll refer to it in reference to the genome, which describes all of the information kind of collectively. It's sort of like the difference between a paragraph in a book versus the entire book itself. Gene is like a paragraph, genome is like the entire book. But that term is sort of relative, but describes kind of what the information in the cell looks like. Then phenotype is often used to describe how it looks or what are the characteristics that result from that information, whether it's an observable trait or a cell function. One way or the other, the term phenotype kind of gives us an idea from what the DNA is doing, rather than just how the information itself is structured. So this gets into the next lecture when we look at the way DNA is expressed. And that's the next lecture is called gene expression. So in other words, what does the DNA do? How do we understand the nature of what that DNA is coding for? That's kind of the idea of phenotype. So going a little bit further on that, these are some easy examples, kind of like I mentioned freckles earlier. If an organism has dark hair, the phenotype is what we would consider that dark hair. That's a characteristic that we can see, and ultimately we can describe dark hair in reference to the phenotype. Now, if we get down to the genetic level, what we will find is that there is usually at least one, could be more than one gene that actually codes for that dark hair. So we would describe the phenotype in terms of the hair color, but we would describe the genotype in terms of the genetic information that makes the dark hair ultimately manifest. And so in these terms, we can describe differences both at the visible level or at the genetic level is kind of the idea there. So going a little bit further about genes, ultimately genes fall into three categories. So one of the most fundamental that is often discussed the most are called protein coding genes. These are also known as structural genes. The reason they're called structural genes is because if you refer back to previous lessons, proteins ultimately form three-dimensional structures. And three-dimensional structures are what give proteins their function. So when you're describing a protein coding gene, you're ultimately describing how the gene creates protein structure. And so that's kind of how those two terms can be used in a similar way. So these are genes that make proteins based upon the information. Now, as I mentioned, some genes only code for RNA. So not every gene is actually converted into a final protein product. Some genes only get converted to RNA, and RNA is actually the end product rather than it being used to make protein. So these are used in examples such as transfer RNA and ribosomal RNA, and there are many others as well, but these are some of the more fundamental ones. And so here what you find is that these end up taking on a unique role that doesn't necessarily relate directly to protein, like the protein coding genes. So you'll see transfer RNA in particular being discussed in the next lecture on gene expression. And so you'll kind of see there how transfer RNA is used. The last one is arguably the most complex at all, and we don't really get into the details with any of these lessons in this class. But regulatory genes are another major type of gene. And so you can think of these as like the boss genes. So if a protein coding gene is like a tool, then a regulatory gene is a gene that tells that tool what to do. And ultimately, they control how the genes are used. For example, when to produce a protein. So you'll have one gene that makes the protein, and then at least one regulatory gene that kind of acts like a switch, turning it on to make it or turning it off to suppress it. Another aspect of regulation is how much of a protein to produce. Do you need a little bit of that protein or a lot of that protein? That's another aspect of regulation, sort of quantity. And then another key aspect is when or which cells produce it. One of the things you find in the human body, for example, is that you have many different types of cells, and they will all produce a unique set of proteins. So for example, the proteins being produced in your bone cells are different from the proteins being produced in your skin cells or liver cells or heart cells, even though all of those cells have this same information. So what regulatory cells do is they act as in a way to tell the cell which of the genes you're going to produce, when you're going to produce them, and how much of those you're going to produce. And there are other aspects of regulation as well. But those are kind of the big ideas, kind of like the boss genes, do this, do it in this amount, and do it here but not there. And that's kind of the idea of regulatory genes. And those are much more complex. That's really more of an advanced genetics topic. So here we're just kind of laying out some fundamentals. In that particular example, we won't have time to go into the full depth of that. That's sort of a different class in that regard. Well, what we'll talk about next is the structure of genetic material. So to understand how genetics work, you have to actually kind of understand, to at least a fundamental level, what DNA actually looks like in terms of its exact structure. So one of the first things we want to recognize is that DNA, the raw material of our genes, is made up of individual building blocks called nucleotides. And if you recall back from a previous lesson, these are considered a monomer unit for nucleic acids. And the four letters, or the four nucleotides, excuse me, can be expressed as letters. And these are often just in simple referred to as A, T, C, and G. These are actually chemicals, which we'll describe next. But in other words, DNA can be understood fundamentally by recognizing that it's actually made up of four constituent molecules. And these molecules, interestingly, are consistent amongst all life. As far as DNA is concerned, all life forms use these four exact letters and only these four exact letters. And so that's kind of the fundamental essence of the structure, that you have building blocks that are specific to all life, A, T, C, and G. Now, what those actual letters stand for are chemicals that have a more nuanced name. So for example, A stands for adenine, T stands for thymine, G stands for guanine, and C stands for cytosine. Now these are just kind of arbitrary names, but those actually describe the chemicals themselves. So the letters are just a symbol representing a specific chemical that is actually used to build the DNA. Now these are all different chemicals, that's why they have different names, but they all have similar components to them. And we have talked about this in a previous lesson, but to go back over that in more detail here, one of the fundamental aspects of all nucleotides is they have a unit here shown in blue called a nitrogen base. And this is where a lot of nitrogen from our dietary requirements ultimately goes. A lot of it ends up going into building the nitrogen base, which is a key component of every nucleotide. Another key part is called the deoxyribose sugar, shown in orange here. And DNA actually stands for deoxyribose nucleic acid. So that name is in part based upon this specific kind of sugar here. It's not like any other kind of sugar. It's a unique kind of sugar that's only found in DNA. The other key part of a nucleotide is the phosphate group. And sometimes you'll see this shown with just one phosphate. Here you see it shown with 3 phosphates. So they can have 1, 2, or up to 3 phosphates at any given time. And so that part of it is used for energy, and it's used for building the DNA, which requires energy. Now, what you want to recognize next here is that of these 4 DNA nucleotides, they are all the same in the exact way when it comes to the deoxyribose sugar and the phosphate group. Those two, if you were to compare all 4 of these nucleotides, there would be no difference in these 2 parts right here. They would all be the exact same. And that's partly why they're all based in the same grouping, because they have a common core that is consistent amongst all 4 of them. However, the nitrogen base is what differs from one nucleotide to the next. So I'll show that here in the next figure. But this part of it, while you always have a nitrogen base, the exact shape or configuration is unique to all 4 of these. So you can see that here in this figure. This shows all 4 of the letters A, T, C, and G. And if you look at this study, if you study this figure and you look at it carefully, you will find, as I mentioned, that the sugar and the phosphate group is identical on all 4 of these. There's no difference in that whatsoever. But the blue part, the nitrogen base, is different. So if you study that, you'll find that they might be similar in some regard, but they all have unique, exact chemical configurations. So that is the part that makes each 4 different, and the sugar and the phosphate group is what makes them all consistent. Now, the way these letters are used is that they are arranged into what is called a DNA double helix. And some of you have probably heard this term before, or at least aware that DNA has a double helix, something that's not discussed every day, but you hear it from time to time. Sometimes it's described as a spiral staircase kind of structure. You can see that here, what that looks like. What I want to get into, though, is kind of how the units A, T, C, and G build this structure. Now, for simplicity, what most figures will do is they'll kind of lay out the structure in a 2-dimensional form like you see here. So the true form is in a twisted spiral, a helix, but for simplicity, that helical shape is flattened out so we can see it better. And what you find is that the letters make up 2 sides of the molecule, hence the term double, and on each side we'll have one series of letters that then connect to another series of letters on the opposite side. And then they actually twist into that helical structure, as I mentioned there. Now, when we try to break this down further, we can describe the DNA in 2 broad terms. One is called primary structure, and the other one, as I'll mention here, is called secondary structure. What we have in primary structure is a sequence of letters on one side or the other. So, for example, this here, often referred to as a backbone, as you see in this example, A, C, A, C, this part of the molecule can be considered a primary structure. And the other side can also be considered a primary structure. So each backbone is considered a primary structure. And ultimately, that relates to the lettering on each side. Now, more technically, each backbone can be referred to as a phosphodiester backbone. And this is because the chemical bonds that connect one letter to the next on either side are connected by that phosphate group that I showed earlier, and the specific kind of bond that it creates from a chemistry standpoint is called a phosphodiester bond. We don't necessarily have to know exactly what that means, if that helps you understand where that term comes from. So you create a backbone, so to speak, made up of these phosphodiester bonds, and that's ultimately kind of how you get that. So you have 2 backbones, 2 strands, that's where the term double comes from, and they're each made up of a combination of phosphodiester bonds. And those respectively can be referred to as primary structures. Secondary structures result when the 2 backbones connect together. So if you look here carefully, this figure shows little kind of dotted lines that connect an A to a T here, or a C to a G here, etc. Those bonds are what make up the secondary structure. So as it says here, secondary structure results from base pairing between nucleotides across from each backbone in the DNA molecule. So secondary structure connects the two backbones together. So we could describe it from a primary standpoint, looking at just the backbone itself, or the secondary standpoint, looking at how those two backbones connect together. Now, secondary structure results more specifically from hydrogen bonding. As this figure shows here, it's pointing to those little dotted lines. Those are made up of a unique bond called hydrogen bonds. And I mentioned that briefly in a previous lesson where I talked about the role of hydrogen in the cell. Hydrogen does a number of key things, and one of the things that it does is it creates hydrogen bonds. So this is a unique kind of chemical bond, different from the phosphodiester bonds in the backbone, and that's the kind of bond that holds the two strands together. Now, one of the interesting things about hydrogen bonds in this specific context is that the hydrogen bonds only work between quote-unquote matching nucleotide pairs from one to the other. So I'll describe this next here, but if you notice, A only pairs with T and C only pairs with G. So if we look down here, this A is always connected to a T, A, T, and this C is only connected to a G. So those are what we consider matching, or as we'll describe next here, complementary base pairing. So the first question here is, what are the complementary base pairing rules? And the second question is, why is complementary base pairing important? So just to clarify, complementary base pairing rules relate to each of the four letters, A, T, C, and G. And so the complementary letters are always such that if I have an A on one side of a backbone, I have to match that to a T on the other side. And those are reversible. So if I have a T on this side, I always have to have an A on the other side. And if I have a C on one side, I have to have a G on the other side. And vice versa, if I have a G over here, I have a C over here. So A is always complementary to T, and C is always complementary to G in whichever side of the chain they occur on. And so the idea then is that in order to make a hydrogen bond between those two, that hydrogen bond only works properly if I have this complementary base pair on the other side of it. If I don't have the right base pair that matches it, then the hydrogen bond that is being attempted to form there won't work properly. And therefore the DNA structure is said to be abnormal or mutated or altered in some way. So that's ultimately the way that DNA works, is that you have to have matching nucleotides on each side of the strand. Now the next question is, why is complementary base pairing important? And we're going to get to that in a little bit more detail next year. But the general answer, although to be fair, there's more than one answer, but to keep it more specific to what I need you to know here for this, the answer is for proper DNA replication. And so we're going to look at that next here. Proper being the key word there, meaning that in order for it to work correctly, in order for DNA replication, excuse me, to work correctly, these complementary base pairing rules have to apply. And we'll see how that works here in the next part. So DNA replication is a necessary aspect of all life. It's actually a very interesting core requirement of life. It's very complex. All life has a DNA code, and all life has to reproduce in order to propagate itself over time. That's the nature of heredity. Now, the way this works at the cellular level is each cell will grow into a larger structure, duplicate all of the DNA, then divide the extra duplicate copies into equal quantity, and then pass a copy of each set of DNA down to the dividing cells that are the offspring of the original cell. And so the way these terms are used is they call the starting cell the parent cell, and the two resulting offspring the daughter cells. Those are somewhat arbitrary terms, but that's how they're often described. So one cell, the parent cell, grows, divides, and splits into to create two daughter cells. And so in order for each daughter cell to be functional, it has to have a copy of the original DNA from the parent cell. And so what we'll see here is that complementary base pairing allows this to work in an interestingly effective way, because what we end up finding is that the parent cell, the DNA rather in the parent cell, will be used as a template to build the new DNA for the daughter cells. So we'll finish up here by looking at how that works. So if the DNA of a parent cell looks like this, A, T, C, G, G, C, A, T, and then all the complementary strands on the other side. Now in reality, the average cell, just for reference here, an average bacterial cell will have millions of nucleotides. So these are much bigger than just a handful. And a human cell has billions. Okay, so these are very, very large in their totality. But to keep it simple, imagine that the entire genome is represented by just these letters. Now, as the cell is ready to divide, what is actually going to happen is that each of these two backbones are going to be separated. And the hydrogen bonds that make up the secondary structure are going to be split, and the two backbones are separated. So we retain each backbone in each of the daughter cells. But then we can rebuild the other half of it simply by using those complementary base pairing rules. So in other words, with only half the information, we can actually complete the strand in its original form by simply matching up each letter to its complement. And as a result, you can take each strand, separate it, use it as a template, and then build the other half of the strand by following those complementary base pairing rules. So we have half of the DNA in the daughter cells being a direct version of the original parent cell. It's not even necessarily the same information, it's the exact same backbone. So half of the backbone is conserved in the daughter cells. And that's where the term semi-conservative comes from. We're literally keeping that backbone and passing that down to the daughter cells. But in order to make each daughter cell complete, we now have to synthesize the other half from scratch, and it's therefore said to be newly synthesized based on the template strand in each parent cell. So what this ultimately allows us to do is pass on one part of the backbone and then recreate the other backbone. And because we have complementary base pairing rules, this can work very effectively by allowing the DNA from the parent cell to be used as a template for the daughter cells. Now that doesn't even begin to describe the full complexity of DNA replication, but it helps you understand how the nucleotides are put together, how they connect to make a double helix, and then how the complementary pairing allows you to copy the DNA in a highly effective way. Okay, so for this class, we're going to stop there. We don't need to know every aspect of replication, but that kind of gives you a good foundation for the genetics, how it works broadly, how we describe it broadly, and kind of how the structure relates to its function. And next time, we'll get more into how this coding information actually gives us protein and how proteins kind of then are used to make the cellular characteristics themselves.